That's like what I have freshman year.
All right, happy Wednesday. So I just wanted to revisit this if you had a chance to take a look at that and try to kind of name those different mechanisms of the antibiotic resistance. I wrote those in there, what the correct ones should be. There are a few other ones that we talked about that maybe you could think about how to kind of graphically represent that, and that might be helpful. Okay, so also just a reminder, when we meet again Monday, that's our exam 3. Remember, there's homework due up through today, and I'll let you know today when that will be the end of the material. I'm going to try to get through all of 14. I did post some practice questions on Blackboards, I think 12 of them, so take a look at those. Okay, are we good with this? All right, so where we left off last time, we were talking about, we were talking about periods of disease. We also discussed normal microbiota. Sometimes I slip and call it normal flora, but flora makes it sound like it's a plant when it's not. So it's kind of the more accepted way is our normal microbiota or microbiome. We talked about some definitions there. And kind of put a label to periods of disease. So I think the most, probably the newest term there, that prodromal period, remember that's when a patient's symptoms are really kind of mild and non-specific, kind of vague, kind of feel like, uh-oh, I'm starting to get sick. All right, any questions from last time before we move forward? All right, so if we think, so we are going to kind of follow an infection, from kind of start to finish in a host. But thinking about, of course, we've known for maybe 150 years or so that microbes cause disease, right? So thinking kind of historically a little bit, Robert Koch back in the mid 1800s here, he was very concerned with understanding what caused disease. And he's known for having a very meticulous experiments and very well-controlled experiments. So he is most famous for discovering the causation of anthrax, as well as TB. And that tuberculosis, that was a really big deal because that was really prevalent at the time. And up until then, it was assumed that it was a cancer. It's hard to think about that now. Like, wow, we didn't even understand that that was infectious. So he developed these postulates that people used from then on to try to understand what causes a disease. So we call that the etiology of a disease, the causation of disease. Okay, so the way this works, these Koch's postulates are listed here, 1, 2, 3, 4. So the first thing he said is the suspected causative agent must be absent from all healthy test animals and present in all diseased animals. So if he took an animal that, say, died of some infection, whatever, say pneumonia, and he took a tissue sample and he could find, certain microbes in that tissue sample that were not present in a healthy organism. Okay, so he took, and that's not right there. That's not easy to do, to just do, right, a tissue sample and look and see what you can find. But he then said, okay, you have to take that suspected agent and then you have to grow it in pure culture in the lab. Okay, that's another thing that is not as easy as it sounds. You have to figure out the right media and the right conditions to get it to grow up. So you take that suspected agent that wasn't present in a healthy test animal, you grow it up in pure culture in the lab, and then you take some of that and inject it into another lab animal. And if that animal gets sick, and you can maybe see it under the microscope and grow it up again, the same thing in pure culture, then that satisfies the criteria that it's definitely that organism, that bacteria that caused that disease. Okay, so that's known as Koch's postulates, and that had a lot of value for help us kind of systematically understand. Now he happened, like I said, he used figured out what caused anthrax, figured out what caused TB. TB was kind of hard to grow in the lab. Not so much anthrax. He was lucky that he kind of stumbled upon an organism that was easy to stain and see under the scope and easy to grow up in the lab. If he would have been trying to understand some viral infection, he wouldn't have gotten anywhere. Okay, so there are some limitations to Koch's postulates. Besides not being able to culture everything in the lab, sometimes there are some pathogens that are present in people, say, or test animals that are healthy, but they're not making them sick. Okay, so that kind of would screw up that. They wouldn't satisfy Koch's postulate if that was the case. Some things like whooping cough. Sometimes an individual can actually have some of that bordetella pertussis associated with our nasal cavity. Doesn't mean we have whooping cough. We can kind of transiently have it for a while. You may have gotten mono at some point from somebody that wasn't sick from mono, but they just had that virus and they were kind of a carrier. And of course, not all test subjects are going to be equally susceptible to a disease. Some things that can impact that, the role of a person's microbiota. Okay, the kind of different microbes that you have present might help you fight off an infection where somebody else might be more susceptible. We all have slight differences in our microbiota, at least slight differences. And we all have differences genetically in our immune response. And as we look a little bit more closely at our immune system over the next few weeks, hopefully that will be kind of obvious how, or we'll point out some things, how people are genetically different and how we fight an infection can be different. And of course, not all pathogens can be cultured. Viruses have to be cultured a lot differently. You might be trying to discover the cause of a disease that is not even infectious. So, right, something like diabetes or whatever, it could be something that's genetic. Okay, so there are definitely limitations to Koch's postulates. Today, and I'll just kind of touch on this briefly, they talk about something called molecular Koch's postulates in the textbook. Basically, today we're focused more on, we kind of understand in most cases what the actual pathogen is. But now we're kind of leaning more towards trying to understand what's the gene that's responsible for that pathogen causing the infection. So it might be the ability to, make a certain protein. So in the case of this infection that they use, EHEC, that's a strain of E. coli that causes really nasty diarrhea, something that can actually ulcerate the large intestine and cause a bloody diarrhea. Okay, so these are the steps, molecular postulates that we can take to try to figure out what gene is causing a particular, in this case, it's a virotoxin. So we have come a long way since Koch's time in our molecular biology techniques, and we have the ability in some cases to do that. Okay, so I guess just understand here that the goal is to find a gene that causes the organism to be pathogenic. Okay. And I'm not too worried about going through the steps, but if you hear about that, molecular Koch's postulates, that's the goal. All right, so a few more definitions as we move forward here. The capacity of microbes to cause disease. Disease-causing microbes, we call them pathogens. So the capacity of microbes to cause disease, we'll say pathogenicity. And a very closely related word that sometimes we use interchangeably with pathogenicity is virulence. Okay, if we were using these two terms correctly, when we say is something pathogenic or not, that's just a yes, no answer. Yes, it's pathogenic. No, it's not. It's not correct to say, oh, it's really pathogenic. It's more correct to say that organism that's a pathogen has very high virulence or is avirulent. So if it's avirulent, it's not causing disease. So that A meaning not. And if an organism is highly virulent, we would say, maybe we'll say likely to cause disease. And it implies that the disease is serious. Okay, so if something's highly virulent, not only is it likely to cause disease in a host, but that disease is likely to be very serious, meaning life-threatening. Okay. And that's not always the case, but that's we usually measure virulence based on case fatality ratio. So more likely it is to kill you, then that's going to be more highly virulent. Okay, and the traits that a pathogen has that allow it to establish an infection and cause disease in the host, we call those virulence factors. So we're going to get to some examples of those today, things like toxins, things like enzymes that are produced by the pathogen. It's kind of like the arsenal of like a military arsenal, like bullets or tanks or bombs. Those are kind of like the virulence factors, or a way that you could think about their weapons that an organism has. Okay, so different organisms, we can kind of rank organisms based on their virulence. Like some organisms that are highly virulent, that just means, again, that they're more likely to cause an infection, and usually that implies that the infection is serious. So tularemia, that's one that I mentioned. I think streptomycin was first used to treat tularemia. that's an infection that you probably never heard of, but it's called rabbit fever, and it's carried by the fleas on rabbits. So they say don't hunt rabbits in warm weather in summer because you might be exposed to rabbit fever. So all these things that you learn, good information from this class. So that organism is an example of one. Don't memorize that example, but that's highly virulent. The organism that causes bubonic plague, Yersinia pestis, also quite virulent. Whooping cough, less so. Like I mentioned earlier, you can be a carrier of whooping cough and not actually be sick. You can pass it on to maybe a baby that you're taking care of. Pseudomonas, not much less so. If you're otherwise healthy, you're very unlikely to get a Pseudomonas Pseudomonas infection. Burn patients are more likely to have a Pseudomonas infection. You could kind of think of virulence as being contagious. Not in every case. It just means if you're exposed to it, and that may or may not be from another person, but if you're exposed to it, then how likely you are to get the infection. So like anthrax, for example, is in the soil, and that's where its reservoir is. I know I haven't talked about that term yet, but if you are exposed to it in the right amounts, and then it would get you sick. Good. C. diff, another one that's not likely to make you sick unless the circumstances are just right. Same thing with the yeast candida. And then kind of at the bottom of this list, less virulence, lactobacilli. You may have had some lactobacilli for lunch today in your yogurt. Okay, so that's an organism that makes yogurt for us, and we eat it, and it doesn't make us sick ever. Okay, so that's avirulent. Diphtheroides, you probably have those in your large intestine. Okay, so there definitely are differences. The organisms at the top have more virulence factors than the ones at the bottom. Okay, one way that sometimes we measure virulence is by the infectious dose, or sometimes we represent that as lethal dose, because in a test animal, it's sometimes a little difficult to tell if they're sick or not. You can tell whether they're dead or not pretty easily. So for infections that we have good animal models, infectious dose, it's the minimum number of organisms needed to infect the host. So for an example, Q fever, we think it takes one cell of the bacterium that causes Q fever to get you sick. And that actually is not a terribly serious disease most of the time. Cholera, though, for instance, you have to be exposed to 1 billion cells to get cholera. That's fairly easy to do if you drink some contaminated water to ingest that many cells. Sometimes we express this because people are different. The ID 50 is supposed to be the number of bacteria cells to establish an infection in kind of like an average number of animals. So again, animal susceptibility is different, or again, sometimes it's expressed as the lethal dose. Okay, so let's see. The actual infectious dose is going to depend on a couple things. It's going to depend on the portal of entry, which we're going to talk more about portals of entry in just a minute, but that's where in this case, test organism is exposed to the pathogen. So whether that's through their mouth or a cut or whatever. And it also depends on the age, the health, the immune status of the individual. Okay, so the actual numbers of pathogens that you have to be exposed of to get sick, it could be different for different people. And sometimes we have no idea because we don't have a good animal model. But here's just an example of, I think this was from your textbook, some pathogens and their infectious dose, the average infectious dose, we think. So it varies. Sometimes it's as little as one virus particle, okay, norovirus. That's the one that makes you come out at both ends at the same time. It's awful. Salmonella typhimurium, that is salmonella food poisoning, okay? It's a pretty successful organism that only takes one, we think. Giardia, that's the protozoa that causes diarrhea. But then some organisms, it could take, a lot more cells or a lot more viruses. So not to memorize, just to know that there is a big difference. Here's an organism, here's a viral disease that has a very low infectious dose. I've heard that if you had 100 people in a room and they were all susceptible to this disease and one person walked in with this disease, probably 99 of those people would get it. Okay, any guesses from the picture? Red rash, measles. Okay, really low infectious dose, very, very, very contagious. So that's why it's important that we keep our vaccination rates up. Otherwise, it will take off again. Okay. All right. So there's actually two different. Oh, so one more thing. Okay, one more thing I wanted to just point out here. Organisms with low infectious doses are considered more virulent. Okay, so it's one way that we just gauge virulence. If it only takes a few, say, cells of bacteria to cause an infection, we would say that they're more highly virulent. Okay, if it takes a whole bunch of cells, then less so. All right, so there's actually a couple different kinds of pathogens. Okay, a primary pathogen is going to, or sometimes we call it a true pathogen, is going to cause disease no matter, or say regardless of the host health status. Okay, so it doesn't matter who you are or how healthy you are. The healthiest person on the earth can still get gonorrhea, can still get meningitis, can still get a host of other true pathogens, rabies, anthrax. Okay, those are primary pathogens or true pathogens. An opportunistic pathogen, though, you will only infect hosts sort of under the right circumstances. So let's say only cause disease in situations where the host's immunity is compromised. Okay, so an opportunistic pathogen only going to cause disease under sort of a certain set of circumstances. So what are some examples? Say the host is pregnant. Okay, pregnant women have a little bit lowered immunity. That has something to do with our immune system doesn't attack the developing fetus. That's why pregnant women were encouraged to get their COVID shot, because they were a little bit more susceptible to COVID. Burn victims. Okay, if they've lost their portions of their skin is really important to protect us from pathogens. So if that skin is compromised, they're going to be a lot more susceptible to a lot of infections. Any kind of immune suppressing drugs, something like prednisone or some steroid will make you more susceptible to a lot of, kind of dampens down your immune response to a lot of things. Same thing, chemotherapy treatment for maybe cancer. In addition, sometimes an opportunistic pathogen is not always something that we come encounter with. It might be an organism that's already in our body and that our health status changes, and suddenly that pathogen or that organism is overgrowing a little bit. So if part of a microbe in our normal microbiota is introduced into an unusual site, so I'm sure you can probably think of some examples of that. Say a patient's appendix ruptured. Okay, we have lots and lots of different microbes in our large intestine, and our appendix is just an extension of that. If it gets infected and it ruptured, now we have all these microorganisms suddenly in part of the body where they don't belong, and that's a life-threatening circumstance. If you got some kind of a cut, so the cells that are maybe bacteria that are normally on your skin and now get into your bloodstream, okay, that's very bad. That they're going to cause some kind of infection if they get in the bloodstream. Not if they're on the skin, but if they get in the bloodstream. So, okay. So yeah, any kind of stab wounds or anything like that. Okay, there's a certain opportunity arose. All right, so if we look at the stages of an infection, to be successful in establishing itself in a host, a pathogen is going to have to do these things. And there's nothing fancy about these terms. You could call them something slightly different. But the pathogen has to be exposed. I'll refer to us as the host in the right place. So we call those contact points portals of entry. Okay. Oftentimes a pathogen is going to be specific for a certain portal of entry. We swallow HIV, for instance, that's not going to make us sick. Okay, it has to be exposed to us at just the right spot. Typically, there has to be some kind of adhesion for the pathogen at certain kinds of our cells before there can be invasion. Sometimes they're going to invade into our cells, sometimes maybe just deeper into our body tissues. In infection, that just means that if they're going to reproduce and establish themselves, they have to be able to avoid our host immunity. Talk about a few ways that they do that today, more later. And they want to be able to leave that host and go to other hosts. So we call those portals of exit. All right, so let's look at all these steps in a little bit more detail and add in some examples. So many different portals of entry. First thing I might think of as skin, but skin is actually a really effective barrier. We're going to look at that in a little bit more detail probably next week when we start talking about our immune defenses. But the main thing to remember that our outer layer of skin is dead. Okay, so the outer layers are dead cells. Those are continually being sloughed off. So pathogens that might try to invade our skin usually can't attach. and usually going to fall off. So it's kind of rare for a microbe to be able to get into skin as long as it's intact. The most common portal of entry are our mucous membranes. So these are any opening into our body. Basically, they line our body cavities open to the outside. Respiratory tract, GI tract, urinary tract, reproductive tract. Our eyes could be considered a mucous membrane. What do you think is the most common portal of entry, our most common mucous membrane? Respiratory, exactly. Our stomach acid helps out a lot to get rid of anything that we would swallow, but respiratory tract, right, that's the most common one. Our defenses are just a little bit weaker there, I guess you could say. We do have lots of defenses, and we'll talk more about those later. Placenta is another one that's not common at all. There are a few pathogens that can cross that placental barrier. Listeria is one, German measles is another one, but that's not very common. And then sort of a catch-all here in terms of portal of entry, what we call the parenteral route. This word enteron, that's Greek for body, digestive tract, or we'll say Greek for intestinal. Okay, so that's a term, that's a good Greek term to remember. Alimentary tract is another way to say that. So if a root that is par, meaning not, okay, not intestinal, enteral. Basically, this is a catch-all for everything. It's like any portal of entry that's not sort of the normal way. So anything that passes through maybe like our whole digestive tract. So our parenteral route of entry is things that, like maybe a dirty needle that stabs us in the skin and we get an infection from that, or an insect bite, or a stab wound, something like that, like sort of like not the normal way, some particular circumstance introduces that pathogen. All right, so again, like I said, that pathogen usually has to have, it's usually specific for, one of those. And if it's exposed in another way, then it probably wouldn't make us sick. Okay, once a pathogen enters our body through its correct portal of entry, then there has to be some kind of attachment phase. And we have lots of flushing of bodily fluids that tries to keep those pathogens from becoming attached, but they have ways to stick. Okay, so we would call them adhesion factors, surface structures that some kind of microbe might have in order to help it stick to its target cells. We've already talked about some bacterial ones, fimbriae or pili. Again, sometimes we use those two terms sort of interchangeably. Usually if there's a bunch of them all around a cell, we would call them fimbriae. Here's a picture of some fimbriae. Remember, the flagella are used for motility. They're the longer ones, the shorter appendages. Again, most useful for attachment. E. coli, for example, has lots and lots of fimbriae so that it can attach into our the wall of our large intestine. Otherwise, it would just pass out with the feces. So really important to help it stick to whatever tissues that it needs to stick to. Bacterial attachment proteins. Probably the fimbriae or the pili would have special proteins on its surface, so it would recognize the right kind of cell. We have a special name for these attachment proteins called ligands. Sometimes you hear them called adhesins, which is kind of a little bit more intuitive what they actually do, but you hear the term ligands as well. So here's it's always nice to have a graphic to go to that with that. So here's say a bacteria cell. Here's our host cell down here, and these proteins that are probably drawn way bigger than what they actually are. Those are the ligands, and here is some host receptor that the ligands recognize. E. coli, this picture over here, this grayish area, that's showing the E. coli's capsule. Capsules can be sticky and they can help adhere as well. There's attachment proteins that you can't see there, and there's fimbriae that you can't see in this transmission electron micrograph as well. So again, a capsule can be an important adhesion factor as well. If you think about other kinds of microbes, a protozoa like a tapeworm, if you've ever seen, I should look up and show a picture of this, but that head of that tapeworm, they call it a scolex. It has the kind of teeth that will kind of grab a hold of the large intestine. Or even viruses are going to have, we talked about how viral spikes oftentimes, or maybe they'll just have some kind of receptors in the surface. COVID, for example, there's been a lot of press about these ACE2 inhibitors that the COVID, the spikes will recognize. ACE2, that stands for angiotensin, I always forget what that stands for, angiotensin converting enzyme. Those are receptors in the alveoli of our lungs that are important to regulate blood pressure and immune inflammation as well. So the COVID binds to those receptors, and then the patient has trouble regulating their blood pressure and inflammation levels as well. So when COVID binds to that, then we get some endocytosis. That's the method of entry that COVID uses. Okay. So it's enclosing that virus in a membrane and bringing it into the cell. All right, and lastly, there's lots of pathogens that might make lots of different adhesion factors. This is Bordetella pertussis, these little green rods there. That's the causative agent of whooping cough. That organism makes about 8 different proteins that will help it adhere to our ciliated endothelial cells of our respiratory tract. It doesn't actually invade the cells, so it's really important that it sticks there and then it multiplies. All right, so invading deeper into host tissues, some more examples of virulence factors. 3 big ones. We're going to talk about some extracellular enzymes. Also known as exoenzymes. If you're in lab, hopefully you remember we had a whole lab, 5 different exoenzymes that we looked for. We're going to explore the role of toxins. There's a couple different kinds of toxins that we'll talk about, and anti-phagocytic factors. Okay. All right, so in terms of invading deeper into the host or invading into host cells, here's 3 different kinds of virulence factors that are going to be important. So let's look at some examples of each. All right, so the extracellular enzymes that we talked about in lab were all ones that a cell would secrete to break down some macromolecule in order to bring it into the cell to metabolize it for energy. Okay, so here's some examples of how a cell would secrete some proteins, some extracellular enzymes, with the goal of breaking down some host tissue so that the pathogen can invade deeper. So one example here is, excuse me, hyaluronidase. The hyaluron is actually kind of a substance that you could sort of think about as the glue that holds matrix of cells together. The protein, I believe. So that's the hyaluron. And if say a bacteria can secrete hyaluronic acid, then it kind of is able, the bacteria is able to enter in between cells. Okay, another example is collagenase. Okay, that's another extracellular enzyme that collagen is the protein that makes up the connective tissue. So again, breaking down collagenase, breaking down this structural protein, again, for invasion into cell tissues. We'll say degrades collagen. Oh, sorry, hyaluron is a polysaccharide. Said that right. Okay, so sugar that kind of the glue that holds cells together. Here's a few more examples of extracellular enzymes. Here's one that if you are in lab, hopefully this looks familiar, coagulase. This is an enzyme that some cells, it's not very common in the bacterial world, but some of them can secrete coagulase. And what the coagulase does is when it's secreted by bacteria, it helps the bacteria kind of wall off that site of infection. So maybe a patient gets a wound, there's some bacteria in there. If the bacteria can kind of protect itself by clotting the blood around it a little bit, then it's going to be kind of a safe spot. And our host immune cells, our white blood cells that would be attracted to that site, that would try to phagocytize those bacteria, say a macrophage, would not be able to get to those cells. So it allows the cells, like say Staph aureus, to kind of reproduce kind of unhindered. And when the time is right, when that bacteria is running out of nutrients, for example, it will secrete another enzyme, kinase, so it can break out of that clot. And now it's kind of strengthened numbers and it can invade into deeper tissues. Okay, so that's highly correlated with virulence. If you have an organism that can make coagulase, it's probably a little bit more successful in establishing an infection. Is kinase the same thing as staphylokinase? Staphylokinase is the kinase that the staphylococcus will secrete. So yeah, there's different kinds of kinase. There's streptokinase as well. I think it's either the staphylokinase or the streptokinase that sometimes is injected for a patient if they have a heart attack. The first time they have a heart attack, because it can break the clots open. All right, so those are some examples of extracellular enzymes. A couple different kinds of toxins I want to talk about. We have talked about one kind already. Of course, a toxin is just anything that damages host tissues. We have talked about endotoxin already. Okay, so if you remember, endotoxin, also known as lipid A. So if you remember, let me flip to our cell wall structure. If you remember our gram-negative cell wall structure, here's the cell membrane, and then the cell walls composed of those kind of red rods. That's supposed to be peptidoglycan. And then the outer membrane, that's this whole portion out here, that's the outer membrane. And embedded in that outer membrane is the lipopolysaccharide, which is composed of lipid A and 2 core polysaccharides. Okay, so that lipid A, that's the same thing as endotoxin. When is endotoxin released? Well, whenever the cell wall breaks up. So you can see that lipid A is anchored pretty tightly in that outer membrane. When does that outer membrane of the cell wall break up? Well, so that's going to be when the cell divides, maybe to a small degree. You might get a little bit of lipid A released when the cell divides, especially when the cell dies. because whenever a cell dies, that cell wall pretty soon after that starts to break down and be released. Okay, so what does that cause in a host if we have a gram-negative infection? Maybe we take an antibiotic to kill that gram-negative infection, and we have lots of gram-negative cells dying all at once. Well, endotoxin has pretty low toxicity. So that means if we have a little bit of endotoxin release, it's not usually too much of a problem. It causes things like fever, inflammation. We'll talk much more about inflammation another week or two. Some inappropriate blood clotting. Okay, if so on a small scale, this is maybe okay, but if you get a whole bunch of endotoxin release, now the host has a problem. And now if you get too much inflammation, the host can go into shock, and it's potentially be deadly. Okay, so some organisms release a lot of endotoxin and some not as much, but again, this is only gram-negative, because they're the only ones that have that lipopolysaccharide in their outer membrane. No outer membrane for gram-positives. Both gram-positives and gram-negatives can release exotoxins. Gram-positive and gram-negative. These are secreted proteins. Okay, so endotoxin is a lipid. Exotoxins are proteins. So hopefully you're thinking, how are exotoxins different from extracellular enzymes? Okay, both are proteins. Your question? Okay.
Are there toxins in
Oh, so these are virulence factors that are important for invasion. Basically, invasion is part of infection, I guess you could say. So I wouldn't worry about exactly which category. I see what you mean in our 5 stages. So a toxin is going to damage the host. So that's going to give it kind of a leg up in reproducing itself sometimes, and sometimes also invading further. So it actually could be both. I see what you're getting at. Okay. So what I was saying is, how are exotoxins different than extracellular enzymes? The answer is that there's actually a little bit of gray area there. Like sometimes there could be a molecule that could sort of be considered both, but in general, extracellular enzymes are only acting like right next to where the cell that secretes it. So, the cell secretes it and it's acting right locally to it, where an exotoxin is going to have system-wide effects on a host. Okay, so there are secreted proteins that act systemically. Okay, so that means it can act throughout the host body. They have very high toxicity. So in other words, it can kill host cells. Exotoxins kill host cells at low concentrations. Botulism toxin is an example of an exotoxin. I've read that a 6-ounce bottle of botulism toxin would be enough to kill everybody on the planet. Okay, high toxicity. It only takes a smidge to do the trick. And lastly, exotoxins are known for being very specific. Okay, so they might act on only a particular kind of cell. All right, so if we look at some examples of exotoxins, something that's labeled a cytotoxin, that's kind of a general name for a toxin that just kills host cells. A neurotoxin, that's going to be an exotoxin that acts specifically on nerve cells. So botulism toxin is an example of that. Another example, tetanus toxin. We're going to talk more about those later on in the semester. Enterotoxins, another exotoxin. Don't confuse that with endotoxin. It kind of sounds the same, but here's that root entero again. Remember that I said that means intestinal. So these are exotoxins that affect the lining of the gastrointestinal tract. For example, Staph aureus, organism that we're using in lab right now, some strains of Staph aureus make a very potent enterotoxin, so that if food is contaminated with that enterotoxin and we ingest it, Staph aureus is a common cause of food poisoning. Okay, give us diarrhea, make us vomit. And Staph aureus, when we ingest a toxin and it makes us sick, we can start feeling lousy pretty soon after we eat it, within like half an hour. Where some kinds of food poisoning, it might take days and days till the organism grows up in us. But a toxin can start doing its nasty work right away. All right, so here's just a comparison of exotoxins and endotoxins, kind of where they work. I like this comparison table that's in your book. Again, sometimes these are helpful. You have kind of covered up, see if you understand the difference. I would say don't worry about the heat stability. We're not going to worry about that. Lethal dose, again, endotoxin, it takes a whole lot of it to kill somebody, where exotoxin a little bit goes a long way. Okay. All right, questions? All right, so the third kind of virulence factor that a cell might need to, again, either invade deeper into tissues or basically just kind of establish an infection and be successful in making more of itself, has to be able to evade our immune system or have some strategies to do that. So we're going to talk a lot more about phagocytosis, but in general, here's kind of the process of phagocytosis. So say this is the phagocytes is one of our white blood cells, for example. Maybe it's a macrophage. We'll ingest a bacteria and we'll endocytose. Remember, phagocytosis is a kind of endocytosis where it will cover that bacterium in a membrane. We call that the phagosome. And then what actually does the killing is these bags of enzymes called lysosomes that fuses with the phagosome, and all those enzymes and proteins, we're going to talk about some examples of those later, will act on the bacteria. And most of the time it will be successful in killing the bacteria and will kind of exocytose what's left. Interestingly enough, just to kind of set you up for what we'll talk about later, usually a phagocyte is going to present a little piece of what it chewed up from that bacteria. And that's the way that our innate immune cells, like macrophages, will present to some kind of B cell or T cell to elicit a more specific response. Okay, so, but more about that later. All right, so that's phagocytosis. Some bacteria, though, or other microbe, will have mechanisms to try to keep our phagocytes from killing them. We talked about capsule production and how that's one of the reasons that it's so common in the bacterial world for cells to produce capsules, because it makes it harder. It's just kind of, if you think, this is dating me because this movie was a while ago, The Incredibles, Violet in The Incredibles, right? She had this shield around her. That's sort of what how you could think about the capsule protects the cell from phagocytosis. So this is kind of a nice picture here. You can see here's some cells that are encapsulated, and here's one that's not. Our phagocytes just have a harder time kind of A, recognizing the bacteria when they're encapsulating, and B, just kind of holding on to it and enveloping it. So, but eventually our phagocytic cells, like, again, say like a macrophage, will get some help from our adaptive immune system. And eventually they might get some antibodies might be released that will be able to bind to that encapsulated cell. So here we've got an antibody that's binding to that encapsulated cell. The phagocyte will have a receptor for that antibody, and the antibodies will just help the phagocyte kind of get a hold of that bacteria. It's an arms race, but then some bacteria can produce protease. Antibodies are proteins. They'll produce some protease to target antibodies, target and destroy antibodies. So again, it's kind of, well, they do this, and then we do this, and then they have this thing, and then we, it kind of is a back and forth same thing. So here you can see how the protease has broken down the antibody. Some other examples of anti-phagocytic factors, strep pyogenes, which again, if you're in lab, hopefully you recognize that name. We talked about that in lab today, or you will tomorrow. Strep pyogenes has this protein on the surface. And so this is what's called a cell surface protein. I believe your book says the M protein is associated with fimbriae. I do not think that's correct. I've never heard it being associated with fimbriae. It's associated with the surface, with the cell wall of the strep pyogenes. And you can see here a strep pyogenes that has these M proteins, and here's a mutant that doesn't. So you can see them under electron microscope. And what this is just, for now, we could just call it an anti-phagocytic factor. It actually degrades complement, which I know we haven't talked about complement yet, but complement are proteins that our immune system makes that binds to pathogens. So our phagocytes have an easier time phagocytizing them. So basically, the proteins that we make to try to, again, bind to the bacteria cell is not going to be able to bind because of this M protein that the strep pyogenes can make. And also there are a number of organisms that have mechanisms to change their surface proteins. The organism that causes gonorrhea is really good at this. HIV is good at this. The organism that causes Lyme disease is really good at this. So basically, every time we make a specific antibody to bind to the outside of the pathogen, by the time we do that, that takes 7 or 8 or 9 days, it's changed, and those antibodies aren't any good, and we have to make different ones, and then they change. So they change so quickly that they just kind of stay ahead of our specific immune system. All right, so that's just some examples of things that make pathogens successful in establishing themselves. They are always going to, yeah, question. Okay, so explain complement again. I don't want to go too much into that because we are going to talk about it not next week, the week after. But complement proteins are proteins that are circulating in our blood, and they become activated when they encounter pathogens. And one of the main things that complement proteins do is bind to pathogens and just make it easier for phagocytes to kind of grab a hold of the pathogen and ingest them. So yeah, good question, but yeah, okay. We'll get to more of that. All right, so a general rule when a pathogen is leaving the host, as a general rule, sometimes we said it might be only a single cell that enters the host, but as a general rule, they're always going to leave in much larger numbers than what they came. And that's just so they're successful. If we only, if there's only one cell leaving, what's the chances that it's going to infect another individual? So generally shed in large numbers. The portals of exit, you'll notice, are pretty much mirror the portals of entry. A respiratory or salivary portal, we're breathing out even when we talk. We're spraying a little saliva from time to time, certainly when we cough or sneeze. So that's a really common one. One of my TAs in lab, she had a little cold this week, and so she had a mask on. And I was like, you know what? I know she didn't have COVID, but she was just had that mask on so she didn't infect other people with her cold. And I thought, that's really nice of her. Maybe we should all do that going forward. Rabies, if you think about like a rabid dog, you think about the foaming of the mouth. Well, end stage rabies is in the salivary glands, and dogs or humans or whatever, when they're in end stage rabies, swallowing is so painful that they don't bother, and so saliva just kind of drips out, and the virus is in that saliva. Fecal exit. So any diarrheal disease will probably have a fecal exit. They have methods to increase the motility of the bowel. Here's a striking picture. Have you ever seen a cholera cot? Right. So cholera is a bacterial infection that causes people to just that motility of bowel. They just have so much diarrhea that they're so weak that usually they can't even get up to get to the bathroom. So cholera patients, they put on these cots that have holes in them, and they just let the diarrhea run out of them, basically. And the key to treating cholera is to keep the patient hydrated, because they have so much diarrhea that they can die within a day or two just of dehydration. So looks tough, but again, with good treatment, people can survive that. So other portals of exit, your genital tract, semen, vaginal discharge, obviously sexually transmitted infections. Urine is not a common one, but there's a few things like typhoid fever that can be transmitted in urine. Bleeding, of course, things like HIV, lots of other blood pathogens. Okay, so don't leave, but that is the end of the material for Monday. So that's chapter 14. There's some homework with chapter 14. Chapter 15, I pushed off till obviously the next exam. I knew we weren't going to get to that. But chapter 15 is just kind of piggybacking on everything that we just talked about. It's a little shorter chapter, so I do want to keep going here. It's with the pandemic that we've just been through, these are terms that you've probably heard, but a lot of terminology talking about epidemiology and how pathogens are transferred between people. All right, so that term epidemiology, it's become kind of a hot topic. A lot of students are kind of rushing to be part of public health programs. Epidemiology is a study of where and when disease occur, as well as the causation, the etiology. So an epidemiologist, basically, a big part of their job is asking the right questions, as well as the statistical analysis. And then somebody left. Statistical analysis. So a lot of math with epidemiology. I know we ate at a restaurant once a few years ago, and my whole family got sick after we ate at the restaurant. So we called the public health department, and they had a lot of questions for us. What did you eat? What did you eat yesterday? What did you eat before the day of that? What did you do that day? Tons of questions to try to figure out exactly what went wrong. And the restaurant ended up being closed for a few days to clean. So we weren't the only ones. So terminology, incidence and prevalence. Gotta try to keep those straight. Incidence is the number of new cases of disease, and prevalence is the total number of people that have the disease in the population at any one time. Okay, so incidence, new cases. Here's a graph of the incidence and prevalence of HIV in the US. So the HIV pandemic, started early 80s, right? So the blue line is the new infections, the incidence. You can see that it has gone down somewhat from its heights in the early 80s. I'd be interested to see, I should look this up. I bet the incidence of HIV is going up a little bit because all sexually transmitted diseases are ticking up. I think people are not as afraid anymore of getting HIV because we're getting better at treating it. People are living a long time. So that's why the prevalence is so high, because people don't die from it right away. Okay, so you would never have the incidence be higher than the prevalence. Remember, prevalence is the total number of people that are living with the infection. Okay. Some other terms that describe incidence. Sometimes we describe diseases as being sporadic. That just means you get a few cases kind of here and there, not a lot. If a disease is considered endemic and we sort of are living with it all the time, it might have seasonal variations, but it's kind of predictable numbers that we'll have. And anytime a disease is considered epidemic, means we have a higher than usual number of cases kind of right in one time period. And then a pandemic is when we have an epidemic on more than one continent. All right, so how do we know what kind of the normal numbers of disease is? This link, see if I can get it to work, is for the CDC. Ooh, sometimes this doesn't work very well. Oh. Okay, so this is the notifiable disease chart. Whoops, that. Well, this was no, that was not what I meant to do. Sorry, let me try again. I was opening this up and looking at it earlier. So basically there's this list of infections. That's what I wanted. This list of infections that if you go to the doctor and the doctor says, yes, you have this disease, then doctors are by law required to report that to the CDC. So things like botulism or cholera or dengue, gonorrhea, these are all notifiable diseases. Here, let's click on malaria. And you can find out, this is recent, this is for the week ending March 4th, 2023. So this is up to date. You can look up and see, well, has anybody in the US been diagnosed with malaria? Well, currently, as of this week in the US, we had 8 people diagnosed with malaria. This next column, the previous 52 weeks, that's in the last year, the week with the highest malaria count was 66, okay? And then the next column, year to date, 2023, it looks like we've had 206 in the US so far this year. Compare that to last year, we had 232 last year. So we're doing okay. We don't have as much malaria as last year. So, right, this is how we just check and see how we're doing compared to previous weeks. And you can look, I think, let's see, in Michigan, no, we didn't have any malaria cases this week, but we've had and no more than 2 cases of malaria. So obviously these are people flying in from other countries where malaria is endemic. So that's how we kind of keep tabs. And if we suddenly see, oh my gosh, so we got this huge cluster of whatever, then we can kind of keep tabs on that. Okay, so just kind of pictorial representation of an endemic disease. Again, something like colds or flu would be considered endemic. A sporadic infection, again, kind of shows up here and there, maybe something like tetanus, something that's not contagious particularly, just a few people get it now and then. An epidemic, okay, lots of examples of that. Flu can be epidemic as well. We kind of, again, keep tabs on flu numbers as well. And then pandemic, more than one continent. This is a figure from your book that talks about cases, it's deaths due to influenza and pneumonia. So you can see how it kind of goes up and down, right? In the winters, we have more. In the summers, we have less. That doesn't mean that we call it an epidemic every winter. It's a predictable pattern. It's probably always going to be with us. Now you can see here we had a big spike in, looks like 2004. I remember this in 2008. That's the last time that we had really serious flu. We called that a flu epidemic in 2008. And that's significant because typically about every 10 years, we have kind of a big spike. So the last few years, we've been, get your flu shots, because we've just kind of been thinking that it's going to hit hard again, especially with COVID. So this question came up because, right, of COVID. Okay, when do we stop calling COVID an epidemic? When can we consider it endemic? Well, according to the CDC, when an infection is circulating at kind of a predictable pattern, then we might consider it endemic. Nobody wants to talk about it, but we are not there yet with COVID. There's still the potential for mutations that we would get a variation of COVID that our vaccines wouldn't work for, and we would have a ton of cases again. So again, we're not there yet. Hopefully we will be. What also plays into that decision of whether we consider it endemic or not is basically what level people are willing to tolerate. So in other words, is it if we have a lack of social disruption? So you could argue there's, it's causing way less social disruption now. A lot of people aren't wearing masks anymore. It's still around and people are still getting it, but people are kind of more willing to live with it. So I don't know, you could make the case that it, for that reason, that it's kind of on its way to be considered endemic. Okay. I think this is a good place to stop. I have a few other definitions, really, just to close out that chapter. See me after class if you have any questions. Remember, I got lots of office hours at the end of this week. Don't be afraid to stop by if you want to review anything. Have a good day.